Good afternoon all. Today let's discuss about DNA sequencing methods. What is DNA sequencing? Determining the order of nucleotides present in the given fragment of DNA. We all know DNA is composed of just 4 different nucleotides A, T, G, C, right? But we should determine what is the order of these nucleotides present in the given fragment of DNA. So the DNA sequencing is a process of determining the nucleotides order of a given DNA fragment. And why do we need to determine this nucleotide sequence, or why do we need to determine the DNA sequence? It has got several research applications. The first step is knowing the sequence of the given fragment of DNA. If we know the sequence of the DNA, that will help us in further characterizing and analyzing that particular sequence of DNA. And that will help us in diagnosing several diseases. Once we know the diagnosis of the disease, once we could correctly diagnose what is the disease, then that will help us in prescribing the right treatments for disorders, especially in case of genetic diseases. So this slide tells you about what is the DNA sequencing. Why do we need to do DNA sequencing? There are 4 major developments. Because of which, we are able to sequence the DNA. The first development is the discovery of restriction endonucleases. You all know what are restriction endonucleases. In the beginning of this RDT course, we discussed quite in detail about this restriction endonucleases. These are the enzymes which are described as molecular scissors. These are used to cut the DNA, and the products of this restriction enzyme will be specific in their length. And in fact, in today's lab, you have done restriction digestion, isn't it? Practically, you experienced how these restriction enzymes work. If I'm right, you worked with EcoR1 and HINT3. So after the discovery of this restriction endonucleases, we were able to cut the DNA at specific sites and produce the discrete fragments of the DNA. That is one discovery which enabled us to determine the sequence of given DNA fragment. The next development is improvement in gel electrophoresis procedures. We have now improved gel electrophoresis procedures to that extent that 2 nucleotide fragments which are different just by a single nucleotide can be separated by this gel electrophoresis techniques. So the improvement is to that extent, even the nucleotide fragments which are different just by a single nucleotide can be separated by using this gel electrophoresis techniques. And the third development is the gene cloning technique. So with the development of this gene cloning techniques, now we are in condition to produce, isolate and purify the sufficient amounts of DNA required for analysis and characterization. So the third development is the gene cloning technique, by which we are able to sequence DNA these days. And finally, the actual DNA sequencing methods. There are 2 different DNA sequencing methods which are developed, because of which we are able to sequence the given fragment of DNA. I hope you are able to understand. Here I am trying to discuss, there are 4 major developments in the science which enable us to sequence the DNA. Those 4 developments are discovery of restriction endonucleases, and then improvement in gel electrophoresis procedures, development of cloning techniques, and 2 different methods which are available for sequencing the DNA. Now let's concentrate on the 2 methods of DNA sequencing which are available for sequencing the given fragment of DNA. The 2 important or basic methods are MAGSM and Gilbert method. And the second method is Sanger's dideoxynucleoside triphosphate chain terminator method. These are the two methods. Maxam and Gilbert method is also called as chemical method, whereas Sanger's dideoxynucleoside method is also called as enzymatic method of DNA sequencing or the chain termination method of DNA sequencing. But there are certain things that are common in these both the techniques. What are those common features of both these methods? Both these methods, they depend on the generation of a population of DNA fragments, and this population of DNA fragments all have one end in common. Usually the 5' end is common end, and they terminate all possible positions at the other end. That's how a population of DNA fragments are generated. And the population of DNA fragments which are generated in the first step will be separated using polyacrylamide gel electrophoresis method. Once they are separated, but here once they are separated, then there is a method to determine the sequence of DNA. Of course, we expose the gel to the X-ray film. By autoradiography, we will be able to read the sequence. So in both these methods, another common point is that we have to carry out 4 parallel reactions. Each one will generate a set of fragments terminating at one particular base. Since there are 4 bases present in DNA, here we need to carry out 4 parallel reactions, generating a set of fragments in all the 4 parallel reactions. After these parallel reaction products are formed, we have to separate these fragments on polyacrylamide gel electrophoresis. And these 4 reactions, each reaction that generates set of fragments terminating at one of the 4 bases in the DNA, as I told you earlier. And both the methods, Maxam-Gilbert, which is a chemical method, and Sanger's dideoxy method, which is a enzymatic method, both these methods are named after the scientists who invented these methods or techniques. First, let's discuss the principle and the other details of the chemical method of DNA sequencing, that is Maxam and Gilbert method. These are the 2 scientists, Maxam and Gilbert, who developed this technique. In this technique, first the sequence that is to be sequenced is labeled at one end, and that labeling can be done with radioactive phosphate either at 3' end or at 5' end, or if it is radioactively nucleotide you are using for labeling, in that case usually it is labeled or nucleotide to the 3' end. If it is the radioactive nucleotide we are using for labeling, in that case that is labeled at the 3' end, or if it is radioactive phosphate we are using for labeling, in that case it can be labeled either at 3' end or 5' end. This Maxam and Gilbert method, it works for both single-stranded and double-stranded DNA. This method involves base-specific cleavages which occur in 2 steps. In the first step, what happens is specific chemicals are used for base modification. Once the bases are modified by these specific chemicals, then those modified bases are cut by the usage of piperidine. So what did we discuss here? First, the sequence for which we need to determine the sequence is labeled at one end. We can use radioactive phosphate. If it is radioactive phosphate we are using for labeling, it can be labeled at either 3' end or 5' end, or radioactive nucleotide if you are using, it can be, it will be labeled at 3' end. This method works for both single and double-stranded DNA. This involves base-specific cleavage, which will occur in 2 steps. First, the chemicals are used for base modification. Then, piperidine, it cleaves the modified bases and it cleaves the sugar phosphate backbone of the DNA at that modified base. And during this Maxam Gilbert method of DNA sequencing, we need to follow limiting incubation times and concentration of the components in the reaction, base modifying reaction, should be controlled in such a way that it forms the ladder of progressively larger molecules, which means that the cleavage should take place at all possible termination sites. Here it's not termination site. At all possible sites, the cleavage should happen. In the sense, in the first reaction itself, all the bases are cleaved. It shouldn't be like that. In order to achieve the ladder of progressively larger molecules, say for example, G is there at 3 positions. There should be one cleavage at 1G, and there should be another cleavage at another G, and third cleavage at all the 3Gs should not be cleaved in one fragment. So in order to achieve that kind of ladder of progressively larger molecules, we need to control the incubation time and the concentration of the components in the base modifying reaction. So what are the chemicals that are used for base modification before you go for specific cleavage? Here we use dimethyl sulfate to methylate the guanine bases.
Respond. Can you hear me?
Yes, ma'am.
Okay, fine. All right. Because there was a message that my microphone is disconnected. That's the reason I wanted to check. Anyway, let's carry on. Where I was, yeah, dimethyl sulfate is used to methylate guanine bases, and formic acid will attack the purine bases. So when you use formic acid, it will attack either on A or G, and then hydrazine is used to hydrolyze the pyrimidines. It could be C or T. And when hydrazine is used, the high salt that inhibits the T reaction. That means when hydrazine is used, and if the salt concentration is very high, in that case it attacks only C, that inhibits the T reaction. So DMS is used to methylate the G bases, and hydrazine is used to hydrolyze the pyrimidines, whereas in case of high concentrations of salt, the T reaction is inhibited, and 4 lanes on the gel. That means after these reactions are done, base modifying reactions are done, you have to load these reaction products in 4 different lines on the gel that allow the sequence to be determined. So now you look at this gel. So this is the gel, and this is the sequence, hypothetical sequence taken to explain how the 4 different reaction products are formed. And once these 4 different reaction products are formed, each reaction product will be loaded onto the each lane of the gel. You take this sequence. So here 4 different base modification reactions will happen, isn't it? So what did we discuss on A and G? What will act? Formic acid will act on A and G. So one reaction is A and G reaction. So here 3 different products are formed. How 3 different products are formed? If you look at this sequence, carefully observe this sequence. G is there as a first base, and where else you have G? Here, right? Here is another G. So when formic acid is used, it can modify this G, right? And it can also modify, one sec, GCT, GCTAC. Yeah, here another G is there. So GCT. So here one cleavage can happen in that case because the formic acid will modify A and G. So if it modifies A, if the cleavage takes place, one product is GCT, right? And GCTAC. So on this G, if it acts, this will be the product formed. And on this A, if it acts, this is the product formed. So you have to control the incubation time and the concentration of the components in the base modifying reaction in such a way that there should be cleavage here, there should be cleavage here, and also there should be cleavage here. So you will have the formation of all these three different products. This is what I meant. The ladder of progressively larger molecules are formed in such a way you have to control the uh, incubation time and also the concentration of the reagents in the base modifying reaction. Then what is that other reaction? The DMS that modifies the guanine bases. So here there could be guanine and so with the guanine, you will have only one thing, actually one more here also that should have been the product formation. Anyway, if this is not clear also, here what you need to understand is at all possible sites, the cleavage should take place. There should be modification and the cleavage should take place. So if you look at this 4 different reactions where A and G will be modified, where G will be modified, G will be modified by DMS, and whereas C will be modified by hydrazine, C and T will be modified by hydrazine. If high salt concentration is there, only C will be modified. So like this, a set of fragments are formed in the each reaction, 4 parallel reactions, and these are the products formed by each reaction. So once these reaction products are formed, this reaction product will be loaded on each line of the gel. So A plus G gel and A plus G well, G well, C well, C plus T well. So you load these reaction products on 4 different planes on the gel and allow the gel to run. So this is how the bands are formed. Once the bands are formed, since these are radioactively labeled fragments, you can detect them by autoradiography. And you all know in polyacrylamide gel electrophoresis, the principle is that the shortest fragment will travel to the greatest distance. That means the bottommost fragment will be the shortest fragment. So here if you see on the same level, you have band here also, C plus T also, you have band here in C also. If the band is formed in both the position CT and C, that means there the base should be C. That means it is terminated at C. So you write C here. Okay. And then here there is a band in C plus T. If there is band in C plus T, there is no band in C, which means that it is T. It is terminated. The cleavage took place at T. That means it represents the T base. And here you have the band at A plus G well. And you don't have band at G well, which means that this base is A. And here again it is C. Here it is G. Why it is G? Because the bands are there. A plus G well also, G well also. And here it is T, it is A. So now you can read the sequence in 5' to 3' direction from bottom to the top. That will give you the sequence of the DNA which is to be determined. So C, T, A, C, G, T, A. Completely it is not given. That is why here also some variation was coming when I was trying to show the where the cleavage take place. I think one base is missed here. Anyway, this is the principle how you can determine the sequence of given fragment of DNA by this Maxam-Gilbert method. If you have any questions at this point, you can ask me if anything is confusing. Shall I go ahead? Or you want to ask any questions here? One of you please respond. Otherwise, how will I know whether you are following or not?
Yes, ma'am.
Okay. So you are able to follow. Shall I proceed further or you have any questions here to ask?
No, ma'am.
Okay. Okay. So in that case, I will proceed further. Okay.
Okay, ma'am.
Sure. So the next method is Sanger's method or chain termination method. I also told that this method is called enzymatic method because this is here enzymatic termination take place. This Sanger sequencing was developed by the scientist Frederick Sanger and his colleagues in the year 1977. The development of this technique also won the scientist Sanger the Nobel Prize in the field of chemistry in 1980. The development of this Sanger's dideoxy method that has got very major role in the completion of human genome project also, because there the major sequencing was carried out by this principle, that is Sanger's dideoxy method. Though this Sanger's dideoxy method, the original principle, has been replaced by the next generation sequencing method, this remains the primary basic technique even today in many of the smaller scale projects. So here, the main principle is that in Sanger's dideoxy method, it follows the chain termination technique. How this chain termination take place? Here, we all know during the synthesis of DNA, the precursors are deoxynucleoside triphosphates. So for the DNA synthesis to take place, the requirements are, of course, primer that should have the free 3' hydroxyl group. And as per the template sequence, one by one, the nucleotides will be added to this growing 3' end of the chain. So there the precursors are deoxynucleoside triphosphates. But here we use dideoxynucleoside triphosphate. So what is the difference between this DNTP and DDNTP? DNTP, which is the regular precursor we use for DNA sequencing, has free hydroxyl group at the third position. 1, 2, 3. Third carbon will have free hydroxyl group, and this is the group on which the next incoming nucleotide will get attached, isn't it? But here, instead of DNTP, if we use DDNTP, when it comes and gets added to the growing chain, there is no free OH group at the third position of the ribose sugar, I mean deoxyribose sugar. So when there is no free 3-hydroxyl group, the further extension of the DNA does not take place. That's where the chain gets terminated. That is why this method is called chain termination method. Okay, so why it is called chain termination method? We use dideoxynucleoside triphosphate as chain terminators, which will help us in determining the sequence of DNA. So Here also we carry out 4 parallel reactions in order to determine the nucleotide sequence of given DNA fragment. So all the 4 reactions contain the template strand, that is the requirement, template strand in the single strand form, and primer is required. You all know for the DNA synthesis to begin, primer is required, and it also contains E. coli DNA polymerase 1 clenofragment. For the DNA synthesis to take place, always it requires the DNA polymerase enzyme. Here the enzyme what we use is E. coli DNA polymerase 1 clenofragment. Clenofragment is the, when you cut this DNA polymerase 1 with trypsin enzyme, it is divided into 2 halves, larger half and smaller half. The larger half is the clenofragment of the DNA polymerase 1. This clenofragment of DNA polymerase 1 consists of 5' to 3' polymerase activity, and it also contains 3' to 5' exonuclease activity. So what is missing here is 5' to 3' 5' to 3' exonuclease activity, and missing this 5' to 3' exonuclease activity is very critical for this technique. Why? Because in the beginning, if you remember, I told that all the fragments that are generated in these 4 reactions will have one common end, right? Since it does not have any 5' to 3' exonuclease activity, the 5' ends of these generated fragments that remain same. Otherwise, the primer 5' end, here 5' end in the sense the primer sequence, that would have got degraded by this 5' to 3' exonuclease activity of polymerase 1, then the whole principle of this technique doesn't work. Okay, so here the point is that here also we carry out 4 parallel reactions, and all the 4 reactions will have template strand of the DNA. That means the fragment for which sequence needs to be determined, and the primer is required. E. coli DNA polymerase 1 clenofragment, the enzyme is used, and the precursors of DNA, all 4 DNTPs are used, DGTP, DATP, DTTP, and usually one of the precursors will be radioactively labeled. Okay, in addition, each of the 4 reactions, so till here only one reaction mix we can have. Once all these components are added, then we can divide the reaction into 4 different parts, and each part we will add one of the 4 chain terminators. What are this one of the 4 chain terminators? One reaction will have DDGTP, the second reaction will have DDATP, third reaction will have DDTTP, and fourth reaction will have DDCTP. See, this is how here we do this chain termination method of DNA sequencing. Single strand DNA of unknown sequence for which we have to determine the sequence. I told you one of the precursors can be radioactively labeled, or sometimes the primer what we use can be radioactively labeled at the 5' end. So in this particular example, the 5' radioactively labeled primer is used. Primer. So all the 4 precursors need to be added. That is all the 4 precursors in the sense, what are they? DATP, DGTP, DCTP, and DGTP. All the 4 precursors. And after all the 4 precursors are added, you divide that reaction into 4 different parts, and each reaction will have one of the chain terminator. So this reaction DDATP, this reaction DDCTP, this will have DDTTP, and this will have DDGTP, right? So these are the 4 parallel reactions. So after they are divided into 4 parallel reactions, you can allow the denaturation to take place, and after denaturation, you can separate them by gel electrophoresis, Okay, so this is how we can separate them by gel electrophoresis. So before this step, what you need to understand, so here it is shown, once you allow the reaction to take place, so here it is not mentioned, in this reaction, another component that should be present is DNA polymerase and, okay, here it is mentioned, DNA polymerase and DNTPs. So in the reaction, what are all there? Template, primer, DNA polymerase for the polymerization to take place, and DNTPs, which are the precursors. Then the reaction is divided into 4 parts. Each part has one chain terminator. This is dideoxynucleoside triphosphate. So here the structure is given for dideoxyadenosine triphosphate. As I told you, at the third position of the deoxyribose sugar, there is no hydroxyl group. That is how it terminates the reaction when this is added to the growing chain of the DNA. So once the reaction is allowed to happen, you see how the products are formed. So here it is shown the reaction products, how they are formed. This is the sequence, right? You look at the sequence in 5' to 3' direction. This is the sequence. So we are talking about the reaction where the chain terminator is dideoxyadenosine triphosphate. That means there is possibility that this DDATP gets incorporated during the synthesis of DNA wherever there is T in the template. Am I right? Wherever there is T in the template. So how many positions there are T in the template? Just look at it. Yeah. 1, 2, 3 positions. There are 3 positions in the template where T is present. That means there are 3 possible termination sites in the reaction where we have taken DDATP as chain terminator. So primer attached here, DNA synthesis will be going on by using the DNA polymerase enzyme and DNTPs. It goes on. Here, this is one possible termination site. So this will be one reaction product. So when DDATP instead of DATP, see, you all should remember that in the reaction there is also DATP. So here at this position, there is possibility that DATP could be added or DDATP. If DATP is added, it grows further. And here another there is possibility for the DDATP or DATP. If DATP is added, it grows further till here. And here another there is possibility. Okay, so here it can get terminated. So here if ATP is added, this will be one product. See here, this is one product. Suppose here DATP is added, then here another possibility, another reaction product that could be formed. If in these 2 positions DATP is added, further it grows, and here if DDATP is formed, then this will be the reaction product. So 3 possible reaction products are formed, and all the 3 will be formed if you maintain the limited incubation time, and if you take care that all the reaction components are maintained in right concentration. In order to get the formation of all the 3 reaction products, we have to maintain the ratio of DNTP and DDNTP in the reaction in 100:1. That means the normal precursor should be 100 molecules and your chain terminator should be 1 molecule. If you are able to maintain the ratio of this DNTP and DDNTP in 100:1, then there is possibility that at all possible termination sites, the chain termination will take place. So only for one reaction it is shown how the reaction products are formed. Similarly, for DTCTP also, how many DTCTP means it is incorporated wherever there is G in the template. So how many positions G is there? Let us see. 1. Right? Only 1. Am I right? Only 1 or more? G. No. G. Yeah. C is incorporated wherever there is G. So I am looking for G. Yeah. Here is one G. Right? G. 2, 1, 2. Only 2 are there, but here I think 3 bands are formed. Anyway, that's fine. G, 1, 2, 3. Maybe here one more G is there. Okay, one more G is there, which is not shown. And then third reaction is DDTTP. DDTTP means where the DDTTP gets incorporated wherever there is A. How many A's are there? 1, 2, 3, 4. Right? Then 4 possible products are formed. DDTTP, right? TTP 1, 2, 3, 4. Actually, this gel and this sequence are not matching, but anyway, that's how it should be formed. So, but here it is explained correctly. So, likewise, in DDGTP also, wherever there is C in the sequence, all those positions, the termination will take place. So, once all these reactions are over, then denature them and separate by polyacrylamide gel electrophoresis, and then expose the gel to the X-ray film. Then you will have the bands formation like this on the autoradiography. And you all know the shortest fragment will move to the longest distance. And once these bands are formed, you can read the gel from the bottom to the top, right? That means this is the fragment which is terminated at C. Okay, the shortest fragment terminated at C. And this is the fragment formed by termination at G. Fragment formed by termination at A. Again A, G, T, C, right? C. And then here it is C, A, G. So when you read the sequence this way, that will give you the nascent fragment. That means the newly synthesized DNA fragment sequence in 5' to 3' direction when you read the gel from bottom to top. And the complementary sequence will be your template sequence for which you want to determine the sequence. The complementary will be C, T, G, and so on. Right? So when you read the gel from bottom to the top, that will give you the nascent fragment, the largest formed nascent fragment in 5' to 3' direction. The complementary sequence will be the sequence of the template strand for which you want to determine the sequence in 5' to 3' end. So that's how you can determine the sequence of the DNA by this Sanger